everyone. It is with my distinct pleasure to be your moderator today. I'm Susan Gilmore. I'm a pediatric hepatologist and transplant physician in the Department of Pediatrics and a longtime member of the ATI. I am really pleased to be able to introduce to you my colleague, and I'd like to say friend as well, Dr. Diana Mager. I will first start with our land acknowledgement. I wish to acknowledge that the University of Alberta is located on Treaty 6 territory and respects the histories, language, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. I also wish to thank Paladin for their ongoing support of the ATI Educational Seminar Series, and also to put the reminder into everyone that the ATI Research Day is happening next week on May the 10th, and there'll be no ATI seminar next week to make room for this wonderful lineup of very informative speakers. This is the final week to register, and we will have it in the chat for you, if you have not already, to register for the ATI Research Day. So by way of introduction, Dr. Diana Mager is a professor in agricultural food and nutrition sciences in the Faculty of ALS. Dr. Mager has conducted for many years research into liver disease and the post-transplant liver disease in children and also in adults. Dr. Mager received her PhD in nutrition at the University of Toronto with Dr. Eve Roberts, and then went on to complete her postdoc with Dr. Paul Penchars, also at the University of Toronto at the Hospital for Sick Children. Dr. Mager was then a scientist with the Hospital for Sick Children prior to her relocation to the University of Alberta. I am pleased to say Diana and I have worked together both clinically and also professionally with students and in research, going back to when we were child prodigies, I told her, in the mid-early 90s, on the wards at Sick Kids, caring for the children there. Her devotion to this patient population has led her on a very productive and informative research career. And today, Dr. Mager is going to speak to us today about the skeletal muscle changes that are found in children who have chronic end-stage liver disease undergoing liver biopsy. Dr. Mager.
Thank you so much for this opportunity to give my talk, and I really appreciate that. And yes, as Dr. Gilmore said, we've been working together for a long time, and I really enjoy working with her and the pediatric transplant team on this project and many others. So the objectives of the talk today are to become familiar with skeletal muscle morphology in children with end-stage liver disease at the time of liver transplant, and to understand what the differences are in skeletal muscle morphology in children that have sarcopenia. And finally, I want to translate that a little bit to then what should be, or what are some of the thoughts about how this might impact the types of exercise therapies that may be beneficial to combat the skeletal muscle changes that occur in children with sarcopenia after liver transplantation. From the point of view of sarcopenia, this is an adult concept that's only been recently identified in pediatrics, and liver disease has been one of the populations where this has been studied. So while we traditionally know it, I think of sarcopenia as a disease of the aging, where there's a decline in skeletal muscle mass, which can result in changes in muscle function and muscle strength. The definition in pediatrics hasn't been well defined. Most of the literature has focused on establishing the fact that myopenia exists in children, but it really doesn't consider some of the unique details of growth, gross motor and fine motor development, and neurocognitive development. And Susan and I and my students have been looking at these different variables to see if we can establish a little bit a better understanding with this. And so one thing that we need to keep in mind is, with the challenge of pediatrics, is children are still accumulating skeletal muscle mass. And so when we think of pediatric sarcopenia, we might need to think a little bit about decreased skeletal muscle mass accumulation, so that the rate of muscle mass development may be impaired, and that can translate to changes in muscle strength, physical performance, and so forth. So when we look at some of the work we've done earlier, we were able to show, for example, that myopenia was fairly prevalent at time of liver transplant. At least a third of the children had reduced skeletal muscle mass. And we've been looking at this in combination with CT and MRI scans, similar to what in adults is done. And you can see from this slide, for example, in the gross motor skills, that the children who had myopenia had significantly lower motor skill percentiles at time of liver transplant versus those that did not have myopenia. And so we know that myopenia definitely exists at time of transplant, but we also wanted to look into the post-transplant period to see if reduced skeletal muscle mass and changes in the ability to perform activities of daily life also occurred in the post-transplant period. And indeed, we were able to show that in children up to 10 years post-liver transplant, that they had some alterations in their muscle functions. So things like sit to stand, as you can see being done here, was significantly lower in children who had undergone liver transplant versus healthy controls in red. They had lower number of push-ups they could perform, and their stair climbing tests were a lot slower. Interestingly enough, that the skeletal muscle deficits were mostly in the lower limbs, which kind of makes sense then why children may experience more difficulties in terms of performing these activities of daily life. So when we looked at this population of the kids between 6 and 18 years, we saw that about 55% had some kind of deficits after 10 years, whether it was in muscle strength or physical performance or in skeletal muscle mass. They had at least 1 to 2 of these components up to 10 years post-transplant. And that made a lot of sense to us because lots of the children would complain in clinic that they had early fatigue with playing in the playground, running, jumping, doing all the things that school-age children do. They just felt more tired. And so chronic fatigue is an issue in this population. And we can see that only 18% of the children in our cohort were within normal healthy reference ranges for all 3 components that have been discussed in the sarcopenia literature. And so we also came to realize that there's a lot of, besides how to define sarcopenia, there's a lot of gaps in the literature and in adults about what kind of changes in skeletal muscle morphology translate to these deficits in physical performance and muscle strength. And so that's when we started to get interested in trying to establish, is there a link between skeletal muscle morphology and pediatric sarcopenia? And then at the end of the rainbow, what would our thoughts be about what would be the best or effective rehabilitation strategies to deal with this? So as you probably are well aware, human skeletal muscle is composed of a variety of different fiber types that are characterized by different myosin heavy chain isoforms. And so when you can see in this table, we can see that the type 1 type of fibers, which are more highly prevalent in healthy children than compared to adults, are the slower oxidative fibers that have lots of mitochondria, and they're very sensitive to inactivity, disuse. Some like to think of these muscle types, type 1, as the type that are slow to get started in terms of energy generation and movement, but they're the ones that are going to help you maintain that activity. Compared to the other type 2 type of fibers, which are glycolytic. So they're faster. The type 2A is fast oxidative with even more mitochondria. And then there's some hybrid forms and types of type 2 that have fewer mitochondria. And those seem to be, instead of being the ones that are associated with inactivity, those are the ones that seem to be in the adult literature very sensitive to the presence of inflammatory conditions like sepsis, cancer, fasting, long-term starvation. And so this is a little bit about our thought process, was maybe the type 2 would be lower in the kids with end-stage liver disease at time of liver transplant. And that's what we wanted to explore. We wanted to see, is there a specific difference in these? And then thinking in mind that the difference between being a slower but lots of energy, slower to start versus a faster muscle, maybe we should gear some of the rehab to address the deficits that could occur. So when we look again, we see in healthy children, muscle fiber types in healthy children are very different in terms of proportions than adults. And so children tend to have, you know, a much higher, not much higher, but a higher level of type 1 and type 2A fiber activities. There's some differences that have been reported with sex. And as we age, the proportion that adults have tend to get more of the type 2. So we do think that it's important to pay attention to this difference between pediatric muscle fiber characteristics or profiles compared to adults, because maybe different strategies for exercise rehabilitation would apply. So children aren't necessarily small adults, and so our approach may need to change depending on this kind of relationship. And we also know that sarcopenia may lead to alterations besides in muscle fibers and the percentage of those, but also the kind of fat and the amount of fat and the type of fat that's present in, for example, within the actual muscle cell or between muscle cells. And so we're also very interested in understanding the fat content, because myosteatosis in adults, for example, with liver disease has been associated with worst preoperative and postoperative outcomes. And we think that understanding the amount of fat and the type of fat, along with the fiber structure and the fiber type, can all be very, very important. So the concept of a fatty muscle that may not be favorable in terms of the profile of different amino acids is something that we're also very interested in these series of studies. So what I'm going to present today is some of our preliminary findings that describe some of the differences in muscle fiber type and proportion. And we did hypothesize that there would be some differences with children with sarcopenia versus non-sarcopenia. And we utilize a variety of techniques to examine this. So here is the MR, and we use MR and CT scans to measure skeletal muscle mass indexes. We measure them at L3 and L4, and we use age, sex matched healthy controls who underwent abdominal MR or CT for investigational purposes as outpatients as our healthy controls to establish the cutoffs. And of course, we need to correct the skeletal muscle mass index by or create an index divided by height squared to normalize for changes in growth with kids. And so we define a myopenia as someone who has a Z score based on that as less than -2. When we look at the muscles, we collect the rectus abdominis muscle to do fiber analysis at time of liver transplant. So we're very fortunate that the surgeons are willing to share with us and give us a muscle, very tiny muscle biopsy samples from the rectus abdominis. We do a variety of histological methods on that, including using laminin and dystrophin to look at cell membrane for quantification of muscle fiber size. And then we also do use immunofluorescent to identify the fiber type. And so that in this particular muscle, the yellow color is the type 1 fiber and the blue color is the type 2 type of fibers. Sometimes we have hybrids present as well. Adults and children can have hybrid formations, and then the blue wouldn't be so distinct from the yellow. And my student Amber tells me that it's not as pretty as this would look when it contains a lot of hybrid. So this picture doesn't contain a lot of hybrid fiber types. We've recruited about 27 patients so far, and of course, unsurprisingly, the main indication for liver transplant was biliary atresia, but you can see we have a variety of other patient populations, such as those with autoimmune disease, PFIC. You'll probably ask me, aren't you concerned about autoimmune diseases if they're on medications such as steroids, which can affect protein turnover in the muscles? And the answer is yes, but our patients at time of liver transplant assessment were not on high dose steroids, though I do acknowledge that they were on low dose steroids. And we only have 2 patients in our cohort, but that's something for us to think about as we continue to recruit for the study. About 62% of them underwent living related transplants. And if we look at the data in terms of how old they were, the children on average at time of liver transplant assessment were 1.7 years, but you can see that many children were about 6 months when they were assessed, and then about 1 in 4 years on average when they went to transplant, but it ranges between 1.3 and 8.4. It's not really surprising to me that as they progress towards liver transplant, that their height Z scores worsen with time. That's a reflection of their challenges with nutritional status and being fed, and their worsening of their liver disease. So we do see some differences in height Z and height between the time of liver transplant and liver transplant assessment. And at transplant assessment, their PEL scores were about 15. When we look at the MR imaging, we can see that we look at a variety of different muscles. The child on the left, the red muscles are the abdominal muscles. You can see it's very compressed and they have myopenia compared to the healthy control, the other liver child without sarcopenia that has a nice robust amount of red skeletal muscle. We do see differences, for example, in subcutaneous adiposity. Many kids have low levels of subcutaneous fat in combination with low skeletal muscle mass, and that's a finding we find fairly consistently, whether the children are 2 months to 4 years of age or whether they're older, that we do see quite a bit of subcutaneous low levels of subcutaneous adiposity. So about a third of our patient population in general has myopenia at time of liver transplant. They tend to be the older kids. In general, our data suggests that they're the kids over 4, and male children tend to have a higher amount of low muscle mass. And that can be partially related to the fact that if they were older at time of liver transplant, they might have had their chronic disease for a longer period of time. And so we're not really surprised by that finding either. When we looked at the children in our cohort that we've collected so far, we see that once again that the myopenic children are a little bit older, as I mentioned. There was a difference between the myopenic and non-myopenic for weight Z scores. Some of that might simply be due to the fact about ascites and the amount of extra fluid they have on board. And then you can see that there's a difference between the myopenic and non-myopenic at time of transplant. When we look at the skeletal muscle fiber isoforms, then we see that the non-myopenic children have more type 1 fibers. It's about 35 to 40% higher amounts of type 1 fibers compared to those that have myopenia. And so that might have some implications about the type of exercise interventions we might want to consider. We also see that there's a difference in the type 2 AD. Some of these influences are affected by age. And so I also examined the data above and below the median age of this whole cohort. And we saw that the children who were younger had more type 1 than the children that were older. And so that makes sense as well to me, because again, it's the myopenic children who tend to be older, who had their liver disease for a longer period of time before they went to liver transplant. So just to show you again, this is an example of a skeletal muscle. You can see in yellow, there's a lot of yellow on the one to the right. This is a patient who did not have low skeletal muscle mass by CT. And then, so lots of yellow type 1 fibers, but the one on the left, you can see a lot more blue and a lot less type 1 fibers. So there's a distinct difference in the amount of type 1 in the myopenic patient. So remember, I want you to remember those type 1s are the ones that have lots of mitochondria, but they're, the slower type of muscle. We also see that there's some differences in centralized nuclei in this. You can't really see it from this picture, but centralized nuclei is an abnormal finding in skeletal muscle. Most nuclei in the muscles are as you can see in this picture, they're at the membrane level. And atrophy is associated with an increased progression of the nuclei becoming centralized in the skeletal muscle. So, some of the philosophy of thoughts behind that is that you need the nuclei to be at the membrane level, so you can get the transfer of oxidative substrates and the exchange of nutrients, and that it's the powerhouse, and that better communication occurs. So when it's centralized, these muscles may be don't function as well. And the presence of centralized nuclei has been associated with atrophy and some conditions of myopathies as well in children with other types of diseases. So what the meaning of a higher level of centralized nuclei in children with myopenia is not really understood, but this is coinciding with what's being shown in the adult studies that are undergoing in Dr. Montana Loza's lab. So a very consistent finding that we see more centralized nuclei in those children that are myopenic. We also see a little bit of differences when we look at skeletal muscle fiber surface area as well, and we find that there's more type 2AD hybrids in the myopenic patients. Didn't find a lot of differences in the type 1 in terms of the surface area. Did find some age differences when we adjusted things as well. We can see that children less than 3.8 years had higher levels of the hybrid type than the older children with myopenia. So there's some distinct differences in the proportions, the presence of centralized nuclei, which are important to consider when you're considering the different types of exercise, because that's the other part I'm going to talk about is rehabilitation. So again, lower type 1 muscle fibers that are slow, have lots of mitochondria, therefore low force production. They're like the types of muscles that are going to carry you through everyday activities. And if they're lower, that probably explains a little bit why the children complain of early fatigue from doing it. Activities of daily life in the post-transplant period, because they walk into transplant like this, and for years afterwards they still complain of quite a bit of fatigue and have lower ability to perform activities of daily life. And so this persistency of lower type 1 compared to healthy children may be something that is a persistent finding. We also see that they have differences in the hybrid, and the hybrid kind of takes you into the activities that are more faster muscle activities, and kids with myopenia have lower amounts of these as well. And they're the type of activities that are more for faster activities, running around and doing anaerobic activities and jumping and having fun. So that's something to think about, because now maybe the type of exercise approach that we should take should address some of these deficits, considering some focus on lower force production. Some endurance exercise might be helpful here. When you combine it with the lower amounts of hybrid type 2A, then maybe also some resistance, or endurance exercise. So some medium running, some medium resistance with intensity might be a good approach to exercise rehabilitation. And so keeping this in mind, what we learned a little bit before we saw these results in the pediatrics, we did develop a study that wanted to address and provide effective rehab for the kids post-transplant. And this study is called the STRONG study, and it's a home-based resistance exercise program for the kids post-liver transplant that's performed 3 times per week. And I'm going to talk a little bit about that and show you what some of the potential positive outcomes can be for kids at risk for sarcopenia. So it's a 12-week intervention study. We developed home-based videos of the resistance exercise. And at baseline, we're looking at a lot of different things. We're looking at body composition using MRI, BIA to determine baseline skeletal muscle mass and lean mass. And we're doing the same thing at 12 weeks. We're looking at components or functional tests associated with sarcopenia. So we're looking at 6-minute walk tests, sit to stand, stair climb, and hand grip, so that we can get measures of physical performance and muscle strength. We are also asking a lot of questions about quality of life, physical activity, and then in PEDS, it's really important to involve the parents and the family in the exercise program. So we are promoting both the parent and the child to participate. We're looking at components of engagement of the child, engagement of the parent in participating in this exercise program. And then as we're showing here, we collect information about routine habitual physical activity by asking them to wear Fitbits, and then we provide them a tablet with the videos. And the exercises, as I mentioned, are conducted 3 days a week, usually about 20 to 30 minutes, depending on the age of the child, because we will do more activities for older kids. The targets of the exercises are leg, chest, shoulders, back, and arm, and we progressively increase the number of sets per month so that we can increase the intensity of the exercise, and we're in constant contact with the family as well. And then after 12 weeks, we're looking at the impact again on these outcomes. Important that we decided to do home-based because, as you know, many of our patients live quite far away. The Stollery program covers a huge geographical area. And so when we developed the study protocol, that was why we developed it to be a home-based remote type of study. And they only come to the university for their baseline assessments and at 12 weeks. When we look at the impact of the 12-week RE on the body composition, we see that the visceral adiposity decreased in the kids with liver transplant significantly, about a 32% decrease in visceral fat index over 12 weeks. And there were decreases in the healthy controls as well that were enrolled in this study. But again, this was a much smaller amount. We didn't really see any major changes in subcutaneous adiposity. And when it came to muscle mass, skeletal muscle mass index, we did see a 10% increase specifically related to those abdominal muscles. So the abdominal oblique muscles, we did see a 10% increase, didn't really see an increase in any of the other skeletal muscle masses indexes. When we look at functional tests, we saw that, again, from baseline to 12 weeks, the liver transplant kids, it did experience an improvement in the amount of distance they walked, no difference with the healthy controls. We saw improvements in sit to stand, push-ups in the liver transplant group. The healthy controls did see an improvement in push-ups, but for everything else, their skeletal muscle mass, their other functional tests were about the same. These were kids between 6 and 18 years of age. And I think the mean age is close to about 9.5 or 10 years old of the children. Most kids were more than 2 years post liver transplant. And we didn't enroll anybody who had a recent episode in the last 6 months of rejection. So we can see from this that resistance exercise can result in some improvements in the functional components of physical performance in the kids. We can also see that in terms of that fatigue and the sense of fatigue children felt, that their scores improve. So an increase in the liver transplant scores in the cognitive domains assesses components of fatigue, and those scores improve, meaning the children said that they had less amount of fatigue associated with that compared to what they did before. Parental responses were interesting. I haven't shown it here, but the parents felt more confident at the end of the 12-week period that their child could actually participate in exercise programming. They felt more confident about their child being capable, and they felt more comfortable overall as well. And I think this is important because there is a lot of helicopter kind of approaches. Parents with children post-transplant are often really nervous for their children to participate in routine physical activity for fear of danger of hurting the new organ or hurting the organ in general. So the engagement of the parents was an important part of this process. And I think even though this is just preliminary data, preliminary results from our STRONG study, it does show that the attitudes of parents can make a huge difference in whether children will participate in routine activities. And some of the results suggest that you could also improve functional and physical performance, which could have a big impact on children's quality of life. And I haven't presented the quality of life data, but we are looking at quality of life as well. when you look at this diagram, my graduate student actually made this for a paper that we've recently had accepted. We're looking at mechanisms again for trying to understand and bring it back to the differences in underlying skeletal muscle biology and about whether this will inform rehabilitation strategies. You can see there's lots of factors that affect the liver, affect skeletal muscles, from hormonal differences to reduced energy intake and increased energy needs and the effect of physical inactivity. These are correlated fairly strongly with the changes in skeletal muscle morphology. So these are just preliminary associations where you can see some differences. But I do think the take-home message from the differences we're seeing is that because children with myopenia may have lower levels of type 1 fibers and more of the and lower levels of the hybrid, that maybe a combination exercise regimen may be useful that combines part of, you know, a resistance exercise approach and endurance exercise. And it seems like our preliminary results do support in the actual exercise intervention that this may be the case. Although I didn't present the lipid data, I'm very interested in whether in the skeletal muscle, is the amount of fat greater in the skeletal muscle, and is the class of fatty acids that you might find in the triglyceride or phospholipid portion, are there differences in, you know, N3 versus N6 fatty acids, and whether or not that might contribute to a change in success or morphology of the muscle. And that's something that we have collected the data. We have to do some more analysis on the fatty acid profiles, but we will be looking at that data across the muscle fiber types and see if we can distinguish between those with myopenia and those without myopenia. So just this slide is my last slide, just to try to tie it in from a lipid point of view. You can see that when you have a fatty muscle, that impacts the quality or efficiency of movement associated with a fatty muscle. Athletes, for example, will have a lot more fat in their muscles as an energy substrate. And it's almost like a paradox, because they could have muscles that might have more fat than the person who isn't the athlete. But the quality of the type of fat they might have may be distinctly different versus someone who's experiencing myopenia and liver disease. And so I do think it's important, not only from a skeletal muscle morphology, fiber typing, and the quality of the characteristics of the nuclei, it's also important to think about how that might be influenced by the presence of myopenia, and whether or not it's possible to do any interventions like nutrition support interventions or exercise interventions or combinations thereof that might address some of these differences. And so I think it's really important to understand some of these things before we pursue multiple interventions. But the adult studies that are being done in this area too give me reason to believe that there's some parallel things that would be effective and helpful. And so it remains to be seen how that data pans out as well. And so I guess the take-home message I wanted to give from this presentation, that I think it's important to understand underlying skeletal muscle morphology, because it can influence the success and shape or influence what you might decide to do in terms of developing intervention strategies to address this concern. And of course, I've been talking mostly about a post-transplant period. These kids likely have skeletal muscle morphology just based on their actual end-stage liver disease. And so right now we're collecting the sample at time of transplant, when it's possible to do that as part of the surgery. And we can't be collecting lots of biopsy samples from kids prior to transplant, but it will be interesting to see in other patient populations where maybe that might be possible. So I wanted to just thank everybody that's been involved in these studies. And there, as you can see, there's a lot of people, but I really want to thank my grad students, Amber, for example, my current students, Megan, Eugen, my RAs, Dr. Gilmore for sure, because she's so wonderfully involved. And the two transplant nurses, Cindy and Cynthia, have been really helpful, as have everybody else on the pediatric transplant team, such as Dr. Kawada. And I want to thank the surgeons, because we couldn't do this project without them. And I wanted to acknowledge the radiologist, Michelle Noga, who's been really involved in these studies, Dr. Vera Masaryk, and the PT and OT academics who support my growing knowledge on rehab from a physical activity point of view. So thank you very much. I'm happy to take any questions.
Thank you so much, Dr. Mager, Diana, for just an excellent presentation. As Sean indicated in the chat box, you'd like to raise your hand and to ask any questions.
I can start off with my own take co-moderator prerogative, but I am curious about the impact of parental diet, exercise, and lifestyle habits, you know, and trying to introduce interventions to promote health and wellness in these children at the same time. You did touch on the fact that many of these parents have come through tough periods, you know, having a child and being wary about them, but what about their own habits and things being an impactor?
Well, I definitely think parental involvement are key, especially in the first 12 years of life. Dietary intake is largely influenced by parental involvement. So parental education and support of their health and well-being is really important support. Your approach to things, it still matters all the way through the child's life cycle. But as you probably know, adolescents are kind of special because they don't want to have anything to do with their parents, what their parents do. I have 2 adolescents myself, and they look to their peers. So I think it's also important to consider. That's why I mentioned family. The siblings are really, really important, as well as an influence in this age group, and their peers and their friends. So it's very important to support their community. And if you want them to engage in rehab at home, especially, engagement of that network is really, really important to ensure that you have improved adherence. Without that, adherence can be very low.
Yeah, well, and you touch on the other part that has me intrigued, which is how do you, all the adaptations you have to make to the interventions to work with a 3-year-old versus a 10-year-old versus a 16-year-old, because we're all very, very different individuals in our decision making and our maturity and things through those time periods, and our parents' influence, of course.
Yeah, so the videos are a little bit different for the 6 to 12-year-olds versus the 13 to 18-year-olds. They need to be different, right? They need to be fun and to engage the children in different ways. So they're a little bit different. I mean, the same idea, resistance exercise with resistance bands, but the music, the colors, the length of time, the level of intensity, some of that's been changed. And that's a pilot study. We've learned some things from engaging in that pilot study. And one thing for sure is from feedback from kids is maybe a mixture of physical activity is a good idea. Like dance is a lot of fun you can do at home. And there's a few other things that we've learned that I think for a larger study we might employ. But I do take your point. It's really important to be flexible and to think about different approaches.
Any other questions? I'll just ask one day, although the numbers are relatively small for this question, is there any difference in the underlying liver disease or even the groups of liver diseases, like the cholestatic versus the hepatocellular? Or I mean, I appreciate that we don't have large numbers to be able to.
I think more of the biliary atresia kids do have myopenia, and in terms of the fiber typing, I couldn't really see a difference by liver disease diagnosis, differences that way, but the percentage that, because biliary atresia often are malnourished, I think they have some differences. And I think some of the older kids who've been there longer, I can see, so they may be the autoimmune kids might have some differences there too, but there were only 2 kids out of 27. So it's hard for me to really like pinpoint that for sure, but we did look at the steroid dose just to see if they were on high steroid doses, and it was 0.5 per kilo or less. So I would defer to you about whether you thought that was high dose or not. So yeah, that's about all I can tease out right now. We do have some samples still that we haven't analyzed, which Amber, my grad student, is very involved with this study. She's been outstanding, and she's going to analyze them at the end of May, and we might be able to tease that out a bit more.
It's always sort of a statement rather than a question. It's always an issue for us in pediatric hepatology and transplantation because the numbers are not large, and we do know from a biochemical and a macrofunction perspective that the kids, despite the different diagnoses, end up at the same place clinically and regular biochemistries. And so for that reason, lump them together for many different studies that you and I have done and many others do. But they aren't always the same disease. It's kind of hard.
Yeah, and I think it's important for me to have pointed out, and I probably didn't, was that we exclude patients with urea cycle disorders and other inborn errors of metabolism that are known to affect protein turnover and protein metabolism beyond the thing. So I think it's important that Dr. Gilmore points out that as a result, sample sizes in peds don't tend to be huge. Having said that, there isn't any other study on skeletal muscle biology in kids at time of liver transplant. And the reason this is possible, besides Dr. Gilmore and the team, is because of the surgeons are willing to give us the pieces. And thank you to my student Amber for faithfully, even when it's in the middle of the night, getting up with me, and she goes and processes the sample, and I just come, or first thing in the morning on a Monday morning when it's a living related transplant. So I want to give a shout out to Amber because she's been fabulous.
Yes, Amber, you do need a big thank you. Are there any other questions for this excellent presentation? And I would like to thank Dr. Mager for an excellent and really informative presentation, and her grad students for all the work that they've done on this, Paladin, our sponsor, and for those of you who have participated today. So thanks all very much.
Thank you, everybody.